more than 70% of the surface of our planet is covered by water, and of that oceanic mass, 95% remains unexplored in detail. Crewed missions have been sent to the Moon more frequently than human beings have descended to the deepest parts of the ocean. The Mariana Trench, the deepest known point on the planet, at approximately 11,000 meters in depth, has been visited by people on only a handful of occasions throughout all of history. The explorer Victor Vescovo, who in 2019 completed what is known as the Five Deeps expedition, descending to the deepest points of the five oceans, summarized it clearly before the Royal Geographical Society. 80% of the ocean is unexplored. When you go below 2,000 meters, people say, "There is nothing down there." I would not agree. The reason is that the deep marine environment represents one of the most extreme physical challenges that exist. At sea level, atmospheric pressure equals 1 atmosphere. In the hadal zone, which begins at 6,000 meters of depth, that pressure exceeds 600 atmospheres. At those depths, the volume of air that occupies 1 cubic meter at the surface would be reduced to less than 2 liters. The structure of water changes under those conditions. Its density increases, its thermal behavior alters, and any material not designed to resist those forces implodes in fractions of a second. The windows of exploration submarines made of polymethyl methacrylate, several centimeters thick, deform slightly under that pressure, a deformation that engineers calculate and tolerate with extremely tight margins. At 7 miles of depth in the Mariana Trench, as one of the pilots of the limiting factor, the submarine that carried out the 5 Deeps described it. There are 8 tons of pressure per square inch, trying to destroy everything inside. But beyond the engineering problems, the deep ocean poses a first-order question. In an environment where there is no sunlight, the temperature approaches the freezing point. Pressure crushes any material not adapted to it, and nutrients arrive in extremely small quantities from the upper layers. How is it possible that life exists, and why does that life take forms so radically different from everything we know? The ocean is divided into vertical zones, defined mainly by the penetration of sunlight. The epipelagic zone covers the first 200 meters, where enough light arrives for photosynthesis. It is the most studied layer, the most biologically productive, and the one that houses most of the known marine species. Below it begins the mesopelagic zone between 200 and 1,000 meters, a twilight territory where light barely reaches enough to orient oneself. Here lives one of the most numerous communities in biomass on the planet. The fish and crustaceans that perform daily vertical migrations, ascending at night to feed near the surface and retreating to the dark refuge during the day. This zone also has a more evocative name, the twilight zone, a term that captures precisely its nature as a threshold between two worlds. From 1,000 meters downward, one enters the bathypelagic zone, a domain of complete darkness where the water temperature oscillates between 2 and 4 degrees Celsius in an almost constant manner. Pressure exceeds 100 atmospheres, and the organisms that inhabit here have developed adaptations that challenge biological intuition. Between 1,000 and 4,000 meters, there is also the abyssopelagic zone, where the ocean floor covers most of the planetary surface. The abyssal plains, vast extensions of fine sediment where for millions of years organic particles have fallen from the surface in what oceanographers call marine snow. Finally, beginning at 6,000 meters, the hadal zone starts. Exclusive to the ocean trenches, the most extreme tectonic fissures on the planet. And in each of them, even in the deepest, forms of life have been found. The mesopelagic zone between 200 and 1,000 meters is what scientists describe as the Manhattan of the deep ocean, a cosmopolis of creatures in constant movement, illuminated by the flashes of bioluminescence, and within that territory each night the largest animal migration on the planet takes place, a migration that occurs in the vertical direction and repeats itself 365 days of the year. Hundreds of millions of tons of biomass ascend from the depths toward the surface to feed under the cover of darkness, and return at nightfall before the arrival of dawn. Lanternfish, myctophids, euphorsiids, salps, jellyfish, siphonophores, and dozens of other groups participate in it. The total volume of living matter involved far exceeds that of any documented terrestrial or aerial migration, including the great migrations of African mammals or birds. For a lanternfish or a krill, this daily displacement is equivalent to climbing a mountain to reach breakfast. Food is at the surface because photosynthesis produces phytoplankton there, but remaining at the surface during the day is equivalent to being visible to visual predators: tunas, dolphins, seabirds. The solution that has evolved is to retreat to the dark depths during the day and rise at night to feed. What is still not completely understood is the coordination mechanism. There is no centralized signal. Each individual responds to local gradients of light, temperature, and chemical signals. The result is an emergent collective synchronization at oceanic scale without a conductor. And that migration has a consequence that goes far beyond local ecology. It is a fundamental mechanism in the global carbon cycle. The organisms that rise at night feed on organic matter produced by photosynthesis, incorporating atmospheric carbon captured by phytoplankton. When they descend during the day, they exhale dissolved carbon dioxide and excrete organic matter in the depths, where it remains out of contact with the atmosphere for decades, centuries, or millennia. Recent estimates indicate that vertical migrations transport between 4 and 5 gigatons of carbon per year toward the deep ocean, equivalent to the total annual emissions of the United States. However, the uncertainty in those estimates is enormous, because the total biomass of the mesopelagic zone is itself a very uncertain number. Acoustic methods detect only a fraction of the real organisms, and some estimates of the total biomass of mesopelagic fish are 10 times greater than others. The twilight zone houses the most numerous vertebrate fauna on the planet. The bristlemouth fish of the genus Cyclothone, known as bristlemouth in English, is probably the most abundant vertebrate on Earth. It is estimated that for every human being there exist 100,000 of these fish, which measure half a pencil and are barely known outside scientific circles. Lanternfish, or myctophids, with their frog-like eyes and their photophores distributed in patterns unique to each of the 250 species of the family, are next in abundance. Hatchetfish, small and with an ultra-thin profile, with nuclear blue photophores distributed like runway lights along their silver belly, complete the contingent of small and almost invisible creatures that constitute most of the biomass of this zone. But the twilight zone also produces more disturbing anatomies. The viperfish has teeth so long in relation to its jaw that they protrude outward like curved fangs, and it solves the problem with a special articulation in the skull that allows the jaw to open at an angle of almost 90 degrees in order to swallow prey of similar size to itself. The black dragonfish has teeth more resistant than those of the great white shark, reinforced with nanocrystals that do not reflect light, making its jaws invisible to prey. The barrelé fish carries its tubular green eyes sealed inside a completely transparent head, from where it can rotate them to look directly upward, outside its own skull. And the four-eyed squid has one large yellow eye that looks upward and one small blue eye that looks downward. A visual solution without equivalent in any other animal. The siphonophores of the genus Prior, which inhabit mainly this zone, are colonial organisms formed by highly specialized zooids, each one incapable of surviving independently. Some zooids only swim, others only capture food, others only reproduce, others produce venom. The colony can reach 40 meters in length, which makes it potentially the longest animal on the planet if the colony is accepted as an individual, although that taxonomic debate remains open. In 2020, off the coast of Western Australia, a spiral colony of the genus Apolemia, with an estimated extension of between 100 and 150 meters, was filmed, a living structure that could easily have passed for a portal to another dimension. On land, only a handful of creatures produce light: fireflies, some worms, fungi and bacteria. In the mesopelagic ocean, bioluminescence is the norm. It is estimated that between 70 and 90% of the organisms that inhabit the mesopelagic and bathypelagic zones are bioluminescent. It is, in the words of the researchers who have descended to observe it in person, the most common language on Earth, the most widespread form of communication on the planet, practiced by organisms ranging from unicellular bacteria to cephalopods with complex brains. The chemical reaction responsible involves a molecule called luciferin, which, when oxidized in the presence of the enzyme luciferase, emits photons of light with an efficiency that no artificial system has been able to match. What is extraordinary is that this capacity has evolved independently at least 40 different times in the history of marine life. The convergent evolution of bioluminescence is one of the most documented and least understood phenomena of marine biology. Something in that environment of absolute darkness makes producing light so advantageous that multiple lineages developed it separately, with different molecules and mechanisms, but with the same functional result. Some organisms use it to attract prey, others to communicate with individuals of their species, others to camouflage themselves, others as an alarm to attract larger predators when they are attacked, and some use it in ways that we still have not understood. The deep-sea anglerfish carries on its head a structure called the esker, a filament that ends in a luminous organ. That light is not produced by the fish itself, but by bioluminescent bacteria that live in symbiosis inside the organ. The anglerfish does not generate its own light, it cultivates it. The bacteria receive shelter and nutrients. In return, they produce the lure that attracts prey towards the jaws of the fish. This symbiosis raises questions about how it was established. What mechanism allowed a fish to develop a specific organ to house specific bacteria, and how those bacteria find the path toward that organ in each new generation. The Pacific black dragonfish goes even further. It produces infrared light in addition to visible light, an extremely rare capacity among marine animals. Given that organisms of the deep ocean are generally blind to infrared wavelengths, this fish can illuminate its prey with a flashlight invisible to them while observing them perfectly. Submarine pilots who have descended to the twilight zone describe bioluminescence as something that cannot be reproduced with any camera. When the artificial lights of the submarine are turned off, the water fills with flashes and constellations of light produced by creatures disturbed by the movement of the vehicle. Some explorers who have made these immersions recount that, when briefly turning on the submarine spotlights and turning them off again in the darkness, the creatures respond with a cascade of reactive light that covers the entire visible extension of the water, as if the ocean returned the greeting. That communion according to those who have experienced it, is impossible to film because it is too ephemeral, too tied to the instant in which it occurs to be captured by any external device. The jellyfish of the twilight zone add their own variation to this spectacle. In the interval between 500 and 2,000 meters, explorers who have descended with submarines describe passing through parades of jellyfish of unusual shapes and sizes that have no equivalent in the surface waters where these creatures are popularly known. Jellyfish with bells of luminous colors in yellow and lilac with dozens of bright tentacles. Jellyfish shaped like a crown that pulse like hearts. Jellyfish with the morphology of a balloon from which multiple threads hang. Jellyfish whose bell imitates a wide-brimmed hat or a ghost. Many species of cnidarians of this zone have never been captured or described, because they are too delicate to survive in a net and disintegrate the moment they come into contact with any solid surface. Only in situ observation through a submarine or a deep camera allows their existence to be documented. That means that biodiversity inventories of the mesopelagic zone that are constructed exclusively from captures with nets are inevitably incomplete in a systematic way. The most fragile organisms, which could also be the most abundant in biomass, are invisible to that method. In the Natural History Museum in London, there is a 40-foot tank that contains the most famous specimen of the deep ocean, a female giant squid 28 feet long known as Archie, a shortened form of the Latin Architeuthis ducks. Archie arrived at the museum frozen and took 4 days to thaw. The scientists who preserved her used gas masks. The body of a giant squid is infused with ammonia, a substance lighter than seawater that allows it to maintain buoyancy. An adult giant squid can reach 12 meters in length and weigh up to a ton. Its 8 arms are as thick as an anaconda, and its 2 hunting tentacles, more elongated than the arms, have serrated suckers at the tips that can grasp prey with considerable force. Its eyes can reach 30 centimeters in diameter, the largest in the animal kingdom. For more than a century, the giant squid was known almost exclusively from partial remains found in the stomachs of sperm whales and from carcasses stranded on beaches. It was not until 2004 that the Japanese oceanographer Tsunemi Kubodera photographed one for the first time in its natural environment at more than 900 meters of depth. And it was not until 2012 that the marine biologist Edith Weider, using a luminous lure that imitated the bioluminescence of the jellyfish Atolla vivillae, repeatedly attracted Architeuthis in front of cameras in the Gulf of Mexico and off the coasts of Japan. The resulting videos were seen by millions of people. Until that moment, practically no one had ever seen one of the most iconic animals of the ocean alive and in its habitat. The debate over whether the giant squid is a fierce predator or a shy opportunist remains active. The stomach contents of sperm whales tell contradictory stories. Squid beaks, indigestible, are found in large quantities in the stomachs of these cetaceans, suggesting that sperm whales hunt them regularly. But there are also historical accounts of battles on the surface where the squid seem to win. In 1875, Captain George Drevar described seeing a squid wrapped twice around a whale, finally dragging it headfirst down into the depths. In 2003, a French racing trimaran came to a sudden stop in the Atlantic when a squid wrapped itself around the hull, bending the rudder before letting go. The sailors calculated that the tentacles were as thick as a human thigh. Architeuthis is not the largest squid in the deep ocean. It shares its tank in the Natural History Museum with a fragment of a different species. The colossal squid, Mesonychoteuthis hamiltoni, with a bulkier mantle, tentacles with hooks curved like a cat's claws, and an overall size that can exceed that of the giant squid in body mass. Beaks have even been found that suggest the existence of an even larger, supercolossal squid not yet scientifically identified. H.G. Wells wrote in 1896 that, "In no branch of zoology are we so much in the dark as with regard to the deep-sea cephalopods." More than 120 years later, that statement remains valid. The Vampiroteuthis infernalis, whose name literally means the vampire from hell, inhabits depths between 500 and 3,000 meters, and has such a particular combination of characteristics that taxonomists have not been able to classify it either among octopuses or squids. It belongs to its own order, of which it is the only known living representative. When it feels threatened, it can invert its mantle over the tentacles as if it were an umbrella turned inside out, covering itself with a membrane that connects all its arms and that has spiny filaments inside. But the most surprising aspect of Vampiroteuthis is its diet. Unlike cephalopods that are active predators, this animal is one of the very few known that feeds mainly on suspended organic matter, marine snow, compacted feces of other animals, and dead bodies. Its tentacles have retractile filaments that can extend up to 8 times the length of the mantle in order to capture these particles. It is a scavenger of the depths that uses the anatomy of an active predator to do the work of a passive filter feeder. In 1873, the research ship HMS Challenger extracted from the depths of the Pacific Ocean a sample of sediment that contained something unexpected: shark teeth of monstrous proportions and fossilized nodules of a size that did not correspond to any known species. Those findings inaugurated a debate about abyssal gigantism that would not close for decades, and that is still not completely resolved. The depths of the sea regularly produce organisms of enormous size in zoological groups, whose shallow water relatives are comparatively tiny. The giant squid has already been mentioned. The giant isopod Bathynomus giganteus, a crustacean resembling a garden woodlouse, can exceed 40 centimeters in length, whereas its terrestrial relatives measure millimeters. The Dumbo octopus of the genus Grimpoteuthis has been photographed on the floor of the Mariana Trench at depths of more than 6,000 meters, with dimensions that notably exceed those of its shallow water relatives. The hexactinellid glass sponges that grow on the floor of the North Pacific form individual structures up to 2 meters in height. Models based on the body size of some of these individuals suggest ages between 10,000 and 23,000 years, which would make them the oldest living animals known on Earth. If those estimates are correct, they began to grow during the last glacial maximum, when continental glaciers covered much of the northern hemisphere and sea level was more than 100 meters lower than it is today. However, the direct dating of these animals is extremely difficult because they lack the clear annual markers that corals or trees have, and the estimates are based on indirect models with large uncertainties. The theories proposed to explain abyssal gigantism are multiple, and none is completely satisfactory by itself. The cold temperature hypothesis is the most intuitive. At low temperatures, metabolism slows, growth continues for longer periods, and organisms do not have the energetic incentive to age and reproduce rapidly. The food scarcity hypothesis proposes that in an environment where food is unpredictable, it is advantageous to have a larger reserve of bodily energy, that is, to be larger. A more recent hypothesis combines several factors. Selection favors large size when it reduces predation, increases the capacity for energy storage, and improves movement efficiency in a 3-dimensional environment where searching for a mate may involve traveling great distances. In the depths where encounters between individuals of the same species are extremely rare, being larger may increase the probability that those encounters are detected. Fish that inhabit depths below 1,000 meters do not resemble any fish that can be seen in a fishbowl or in an aquarium. Their bodies reflect millions of years of selection under conditions that have no equivalent on the surface. But the most notable adaptation of abyssal fish is not visible from the outside. It is in their biochemistry. Extreme pressure destabilizes proteins. Under hundreds of atmospheres, the folded molecules that form enzymes and cellular structures tend to compress and deform, losing their function. Surface fish would die in seconds if exposed to abyssal pressures, not because of external crushing, but because of internal biochemical collapse. Abyssal fish have solved this problem by accumulating in their cells a molecule called trimethylamine oxide, or TMAO, which acts as a molecular stabilizer, counteracting the destabilizing effects of pressure on proteins. And here appears one of the most fascinating patterns discovered in deep-sea biology. The concentration of TMAO in fish tissues increases linearly with the depth at which the species lives. It is not an all-or-nothing mechanism, but a fine and quantifiable adjustment. The problem is that TMAO has a limit. Beyond a certain concentration, its presence begins to interfere with other cellular functions. Calculations by the biologist Mackenzie Geringer and collaborators suggest that there exists a theoretical maximum depth for fish, with bony muscular tissue close to 8,200 meters, beyond which the chemistry simply cannot be sustained. That theoretical prediction was confirmed in a surprising way in 2023 when a team filmed what appears to be the deepest fish ever recorded, a species of abyssal snailfish of the genus Pseudolipais, at 8,336 meters of depth in the Izu-Ogasawara Trench in Japan. The record slightly exceeds the calculated theoretical limit, which suggests that either the model still has factors not yet considered, or this species has found a biochemical solution that we do not yet understand. A perfect example of how the deep ocean tends to invalidate its own theoretical frameworks as soon as we construct them. The swim bladder, the organ that most fish use to regulate their buoyancy, is unusable at great depths because pressure would compress it until it collapsed. Abyssal fish have abandoned this organ in favor of other strategies. Many have muscle tissue reduced to the minimum and skeletons with very little calcium, which makes them literally lighter than the surrounding water. Some accumulate low-density lipids in the liver as a buoyant substitute. The result is that their flesh has a gelatinous consistency that is evident in the few specimens brought to the surface in good condition. Many are also almost transparent, with skin so thin that the internal organs can be seen. This transparency is not a camouflage adaptation, but the result of reducing to a minimum all non-essential tissue in an environment where resources are scarce. The tripod fish, Bathypteroi gralator, has developed a completely different strategy. It simply stops. It extends three extremely elongated fin rays that function as legs and rests on the bottom sediment facing the current, waiting for food particles or small crustaceans to be carried toward it. It can remain motionless for hours. The silver hatchetfish of the genus Argyropalicus, by contrast, has eyes permanently oriented upward, tubular and specialized for detecting silhouettes against the faint glow of the surface, while it produces light on its belly precisely calibrated to match that faint luminosity and eliminate its own silhouette when observed from below. Some of these fish can modify the intensity of their ventral bioluminescence in real time as ambient light changes, a fine adjustment that has no known equivalent in any camouflage technology designed by humans. and some abyssal fish have gone even further in camouflage. Their skin is so densely packed with black pigment that it absorbs 99.5% of any light they encounter, appearing even under the spotlights of a submarine as a solid silhouette with no visible detail. Fish of the twilight zone between 200 and 1,000 meters also have visual adaptations that take the biology of the eye to extremes without equivalent in surface environments. The cock-eyed squid, Histiotuthis heteropsis, has one large yellow eye permanently oriented upward to detect silhouettes against the faint light coming from the surface, and one small blue eye oriented downward that detects the bioluminescence of prey and predators approaching from the depths. A single animal with two eyes designed to solve two completely different optical problems, operating independently. The four-eyed spookfish of the genus Bathylusnops has eyes divided into two halves that function as mirrors instead of lenses, directed in different directions. And the barrel-eye fish, Macropinna microstoma, carries its tubular green eyes sealed inside a completely transparent head, from which it can rotate them to look directly upward, outside its own skull. The transparency of the head also allows it to see through the front part of the skull without any obstruction. The abyssal anglerfish has developed the most extreme reproductive solution known among the vertebrates. The males of many species are between 40 and 60 times smaller than the females, do not develop the luminous lure, have extraordinarily developed sensory organs to detect the pheromones emitted by the females, and cannot feed effectively on their own once they are adults. Their only functional purpose is to find a female and reproduce. When a male finds a female, he attaches by biting her. Over time, their tissues literally fuse. The blood vessels connect, the tissues of the male integrate into those of the female, and the male begins to be nourished directly by her circulatory system. His own organs atrophy, including the eyes and part of the digestive system. What remains is a reproductive appendage of the female, capable of producing sperm when her hormones indicate it. In some species, a single female can carry up to 8 males attached simultaneously. This system is puzzling from an immunological perspective. The fusion of tissues between two different individuals should trigger an immune response that would destroy the attached male. Research published recently has found that abyssal anglerfish with sexual parasitism have functionally lost the genes responsible for several components of the adaptive immune system. They have sacrificed the ability to reject foreign tissue as the price to make reproductive fusion possible. That loss makes them vulnerable to parasites and infections that other fish would resist. But in the abyssal environment, where pathogenic fauna is less diverse than in shallow waters, that evolutionary price appears tolerable. In February 1977, the submarine Alvin descended to the bottom of the eastern Pacific, at about 2,400 meters depth near the Galapagos Islands. The scientists on board expected to find the same monotonous and practically deserted bottom that oceanographers had been documenting for decades. What they found was something so unexpected that they initially thought the instruments were failing. Along a crack in the ocean floor, columns of water at more than 350 degrees Celsius were emanating, heated by magma from the oceanic crust, loaded with sulfur compounds, hydrogen sulfide, and heavy metals. And around those hydrothermal vents, there existed a biological community exuberant and completely unrelated to any source of solar energy. There were tube worms of the genus Riftia that reached 2 meters in length. White crabs of the genus Bythagrea moved among them. Mussels and large clams carpeted the bottom. Octopuses and fish circulated around the perimeter. All of this in an environment where the bottom temperature was 2 degrees, the pressure 240 atmospheres, and the darkness absolute. There was no photosynthesis of any kind. The base of the food chain was chemosynthesis, bacteria and archaea that obtained energy by oxidizing the hydrogen sulfide emanating from the vents, converting inorganic compounds into organic matter without any intervention of sunlight. That discovery reformulated one of the oldest principles of biology, the idea that all life on Earth ultimately depended on the sun. Since 1977, hundreds of hydrothermal vent fields have been discovered in all the oceans of the planet, many of them harboring species exclusive to that specific field, with no known distribution anywhere else in the ocean. The marine biologist Cindy Van Dover, the first woman to pilot the Alvin, and one of the people who has spent the most time studying these communities, has described the sensation of observing them in person as being in front of evidence that life has more dimensions than we had conceived. Some of the scientists who study the unique chemistry of alkaline vents of the lost city type, discovered in the Atlantic in 2000, have pointed out that these white carbonate chimneys could be analogues of the environment where life first arose on Earth, and even comparable to environments that could exist on the icy moons of Jupiter and Saturn, such as Europa and Enceladus, beneath their layers of ice. Riftia pachyptyla is probably the most puzzling animal that has been discovered at the vents. This tube worm reaches 2 meters in length and lacks a mouth, a digestive system, and an anus. In anatomical terms, it has no digestive apparatus of any kind. The answer to the enigma of how it can survive arrived when its internal tissues were analyzed, and a massive organ called the trophosome was found, densely packed with chemosynthetic bacteria. The worm does not eat. It is a living container for bacteria that produce the nutrients it needs. Blood vessels carry hydrogen sulfide and oxygen from the external plume of red hemoglobin to the bacteria inside. And the hemoglobin of Riftia is another anomaly. It has evolved to transport oxygen and hydrogen sulfide simultaneously, without one blocking the other. When invertebrates, hydrogen sulfide is toxic and blocks oxygen transport. It is a molecular solution without equivalent in any other animal evolutionary lineage. Beyond Riftia, the vents act as islands of biodiversity on the ocean floor. Each vent field is an ecosystem with unique fauna. Comparative studies show that the degree of endemism among vents of the Eastern Pacific, those of the Western Pacific and those of the Atlantic, is very high. The species of one trench are not usually found in another. But how then do vent organisms disperse from vent to vent if each field is an isolated oasis? The answer involves planktonic larvae that rise into the water column, are transported by ocean currents for weeks, and descend when they settle if they find suitable conditions. This cycle connects communities separated by thousands of kilometers in a slow but real genetic flow that has been detected by comparing the DNA of populations from different vents. The magnitude of that flow and the factors that control it are still the subject of active study. The Osedax worms discovered in 2002 on the floor of the North Pacific add another chapter to this story. These worms feed exclusively on whale bones that descend to the bottom when cetaceans die, and they also lack a mouth and digestive system. But unlike Riftia, they do not depend on vents. Instead, root-like filaments that penetrate the bone house bacteria that degrade collagen and bone lipids. The males of Osedax are dwarfs that live inside the tube of the female, among dozens and hundreds of them simultaneously in a permanent larval state, with the only known function of producing sperm. Dead whales that descend to the bottom create temporary ecosystems of great richness that can last decades. A single dead blue whale can feed a benthic community for more than 100 years. Whale bones are functionally mobile and temporary oases that punctuate the ocean floor like islands of productivity. The hadal zone comprises the 37 confirmed ocean trenches of the planet, with depths between 6,000 and 11,000 meters. The Mariana Trench, with the Challenger Deep as its deepest point at approximately 11,000 meters, was first visited in 1960 by the Bathyscaphe Trieste, with Don Walsh and Jacques Picard on board. It was not visited again by human beings until 1995, when the Japanese unmanned submersible Kaiko descended to the bottom. In 2019, Victor Vescovo descended to the Challenger Deep in the Limiting Factor, a submarine built by Triton Submarines, and set the depth record for a crewed vehicle. Walsh, who was present at the subsequent celebration at the Royal Geographical Society in London as the only living human who had visited the bottom of the ocean before that moment, received a standing ovation. At the bottom of the Challenger Deep and in the depths of other trenches of the Pacific, amphipods, sea cucumbers, polychaetes and bacteria have been found. Not one or two species, entire communities with their own food chains. Each trench is a partially isolated system with its own fauna. Comparative genetic studies among amphipod communities from different Pacific trenches show that populations from different trenches are more isolated from each other than human populations from different continents. That isolation implies accelerated evolutionary diversification, with genetic differences accumulated over millions of years without exchange between trenches. And yet, in all the trenches studied, life has been found, which raises the question of how the first colonizers arrived in those trenches in the first place. The most probable answer involves planktonic larvae of the same ocean currents that transport organisms from vent to vent, in a dispersal process that operates on geological time scales. At the bottom of the trenches, it has also been found that the concentration of persistent organic pollutants, such as PCBs, is between 50 and 100 times higher than in contaminated surface waters. These compounds, synthesized industrially from the mid-20th century and banned in most countries since the 1980s, reach the hadal bottom transported by organic particles and by the bodies of dead animals. Amphipods of the Mariana Trench have concentrations of PCBs in their tissues that would be toxic to many vertebrates of shallow waters, although the amphipods themselves appear to tolerate them through detoxification mechanisms that have not yet been characterized. And microplastics of less than 5 millimeters have been found in sediments of the trenches at 11,000 meters depth, including nylon and polyester fibers originating from the washing of synthetic clothing in households across the planet. The most inaccessible abyss of the planet has been accumulating the chemical footprint of human industrial activity for decades, although the effects of that accumulation on its ecosystems are still practically unknown. The explorer and scientist Alan Jameson, who led the scientific team of the Five Deeps expedition and has carried out more dives to hadal zones than practically any other living researcher, describes the trenches as environments with a character of their own, difficult to reduce to data. In the Pacific trenches, autonomous landers that are dropped to the bottom and wait with bait for hours record communities of amphipods gather in clouds of hundreds of individuals around the bait. In the Challenger Deep, amphipods of the genus Hirondelia and of the genus Alicella gigantea have been filmed, the latter with lengths exceeding 30 centimeters, which represents another example of abyssal gigantism, in a group that in shallow waters rarely exceeds 2 centimeters. The forces that shape hadal biology, including that tendency toward gigantism in certain groups, remain the subject of active debate. descending to the hadal zone requires vehicles capable of withstanding pressures that no conventional material can resist. The limiting factor, the submarine that carried out the 5 deeps, has a pressure sphere of titanium 90 mm thick. Its windows are made of high-density polymethyl methacrylate. Each system is redundant. If one element fails, another can fulfill its function. And even so, the designers and engineers who work on these vehicles describe dive tests as the moment when their heart rate accelerates, because they know exactly what the margins are and exactly what would happen if any of them failed. John Ramsey, the principal engineer of Triton submarines who designed the limiting factor, described that descending in the submarine that he himself had calculated and built was a very different kind of experience from a pleasure dive. The responsibility of designing a crewed vehicle for extreme depths admits no margin of error, and those who do it know it with unusual clarity. One of the fundamental problems of deep ocean research is that any instrument sent to observe it can cause disturbances. Unmanned underwater vehicles, the ROVs, emit vibrations, artificial light, electromagnetic fields, and heat. The organisms of the abyss, which live in total darkness and almost absolute silence, respond to these disturbances with behaviors that are not representative of their normal activity. The fish and invertebrates observed in ROV recordings are reacting to the presence of the vehicle, not acting normally. This methodological limitation is one of the reasons why many researchers argue that crewed dives, despite their cost and complexity, are irreplaceable. The direct connection between a human observer and the deep environment generates qualitative data that no remote system can produce. In 1873, the HMS Challenger expedition extracted from the floor of the Pacific objects that puzzled scientists. Black nodules of irregular appearance, the size of marbles or baseballs, that covered vast expanses of the ocean floor. It took almost a century to understand what they were and how they formed. Manganese nodules are polymetallic concretions that grow on the ocean floor in a formation process that is one of the slowest known in nature. In 1 million years, they may grow only a few millimeters, accumulating atomic layers of manganese, nickel, copper, cobalt, and other metals around a hard nucleus, which may be a shark tooth, a fragment of rock, or any other available solid material. The Clarion-Clipperton fracture zone in the Central Pacific contains an estimated 21 billion tons of these nodules, in an area comparable in size to the continental United States. But the nodules are not inert objects. They are ecosystems in themselves. Organisms such as corals, anemones, sponges and crinoids anchor themselves to their hard surfaces, since on the abyssal plains, that solid substrate is one of the scarcest resources available. Bacteria and microorganisms live inside them, and the community that grows on and around the nodules in the Clarion-Clipperton fracture zone is, according to every expedition that visits it, more diverse and richer than anyone expected. It is estimated that 90% of the animals collected in that area during recent decades are species new to science. Some do not fit into any known taxonomic group. They represent entire branches of the tree of life that had not been described. The oceanographer Craig Smith of the University of Hawaii, one of the leading experts on the Clarion-Clipperton zone, has pointed out that practically every sediment sample extracted from that area contains hundreds of undescribed species. The sediment of the ocean floor is not only the substrate where abyssal animals live, it is itself one of the densest and oldest microbial ecosystems on the planet. The bacteria and archaea that inhabit deep ocean sediment may be active at depths of several meters below the sediment-water interface, in layers deposited thousands or millions of years ago. Their metabolic rates are extraordinarily slow, orders of magnitude lower than those of bacteria in soil or the surface ocean, but they are detectable through modern geochemical and molecular techniques. Some microorganisms in the sediment appear to divide once every 100 or 1,000 years, a generation time with no known equivalent in any other biological system. In 2020, a team of Japanese researchers published a study describing active aerobic bacteria recovered from sediments of the South Pacific deposited 100 million years ago during the Cretaceous period. These bacteria were not in a spore state. They were metabolically active, capable of growing and dividing in the laboratory when nutrients were provided. They had survived 100 million years with the minimal traces of oxygen that diffused from the water column. That discovery expanded the concept of the deep biosphere and raised questions about the temporal limits of life. Some estimates suggest that this subsurface biosphere may contain as much carbon biomass as all the organisms on the Earth's surface combined. The deep ocean is not a peripheral ecosystem, it is a fundamental part of the planet's total biosphere. Hydrothermal vents, in addition to being extraordinary ecosystems in their own right, have strongly revived a hypothesis about the origin of life. The alkaline hydrothermal vent hypothesis, developed mainly by Mike Russell and William Martin, proposes that life arose in cracks in the primitive ocean floor where alkaline fluid emanating from rock interacted with the slightly acidic ocean water of the Hadean Eon. Those interactions would have generated proton gradients similar to those used by modern cells to produce energy through ATP synthase, and the micropores of the rocky chimneys would have served as protomembranes, where pre-biological chemical reactions could accumulate and become more complex. The hypothesis gains ground because it offers detailed explanations about the energy that the first metabolic systems would have needed, and because the discovery that archaea and bacteria from the environment of modern vents contain enzymes very similar to some of the most primitive known supports. The idea that this environment could have been where everything began. Since the Second World War, military forces and scientists have deployed networks of hydrophones in the depths of the oceans capable of detecting sounds thousands of kilometers away. The SOSUS system of the United States Navy has produced decades of recordings that include sounds of unidentified origin. The most famous was recorded in 1997 by NOAA in the South Pacific. A low-frequency sound of enormous intensity with a spectral signature that does not match any known animal or any typical geological phenomenon, which was nicknamed the bloop. The explanation most widely accepted today is that it is the fracturing of large masses of ice in Antarctica, a phenomenon that produces very low-frequency acoustic signals that propagate over great distances. However, the debate is not completely closed, because some characteristics of the sound are difficult to reconcile with that explanation. The deep ocean is full of sounds not identified with certainty, periodic signals without a clear geological correlate, and low-frequency pulses whose source has been actively sought for decades without definitive results. Between approximately 101,000 meters in depth, in many oceanic regions, there exists a layer where the concentration of dissolved oxygen drops to almost undetectable levels. For decades, it was assumed that these zones were practically sterile from the point of view of metazoan fauna. Recent research has demolished that assumption. In the oxygen minimum zones of the tropical Eastern Pacific and the northern Indian Ocean, communities of organisms have been found that survive at oxygen concentrations of less than 10 micromoles per litre, when most animals require at least 100. some fish can experience severe hypoxia during their hours of refuge in the oxygen minimum zone, slowing their metabolism to levels that under normal conditions would be lethal. The molecular adaptations that allow this tolerance are the subject of active research and have implications for medicine, specifically for the treatment of oxygen-deprived tissues in contexts of ischemia. The rate of discovery of new species in the deep ocean remains high. Every expedition to a new abyssal area regularly produces organisms without previous scientific description. The 5 Deeps expedition carried out 103 dives with autonomous landers, collected more than 100,000 biological samples, and identified 40 new species during the time the expedition lasted. Its sonar systems mapped 212,000 square miles of seafloor with high resolution, an area equivalent to the size of France, 60% of which had never been mapped before. And that is the result of a single expedition with state-of-the-art equipment. It is estimated that the total number of species that inhabit the deep ocean may be between 1 million and 10 million, most of them undescribed. Current catalogues record around 250,000 known species. The abyssal plains are probably the environment with the lowest sampling density on the planet. They cover more than 50% of the Earth's total surface and have been sampled in a significant way in less than 0.1% of their extent. Seamounts, estimated at between 30,000 and 100,000 in the world's oceans, each act as an obstacle to ocean currents that concentrates nutrients and generates particular biological communities. Studies on accessible seamounts have revealed very high rates of endemism, with species exclusive to a single mount with no known distribution anywhere else in the ocean. If that rate is representative of the total, the number of undescribed species exclusive to seamounts could be in the hundreds of thousands. Holothurians or sea cucumbers are one of the most visually abundant groups on abyssal plains when observed with unmanned vehicles. Some genera form aggregations of hundreds of individuals that move slowly across the sediment, ingesting large quantities of it to extract the organic fraction it contains. An abyssal holothurian can ingest the equivalent of its own weight in sediment every day, passing it through the digestive system, extracting bacteria, foraminifera, and organic matter, and returning the inorganic sediment to the seafloor in the form of fecal coils. In some areas of the abyssal Pacific, the density of holothurians is so high that they are the most abundant animal by biomass on the seafloor, which makes them the principal processor of organic matter that arrives from the surface in the form of marine snow. Some genera of abyssal holothurians have developed an unusual form of locomotion. They walk on the tips of their spines as if they were stilts, lifting the body off the sediment to keep the sensory organs of the oral tube away from the substrate and detect chemical signals from the layer of water above the bottom. Foraminifera are protozoa that build shells of calcium carbonate with geometries of remarkable complexity, from perfect logarithmic spirals to structures with multiple branch chambers. When the organism dies, the shell descends and accumulates in the bottom sediment. The analysis of those fossilized shells is one of the most powerful tools of paleoceanography. The isotopic composition of the carbonate records the water temperature at the moment when the shell formed, and the relative abundance of different species indicates the oceanic conditions of each period. Radiolarians produce silica skeletons of even greater geometric complexity, with structures in the form of perforated concentric spheres, radiating spines or three-dimensional meshes that inspired the naturalist Ernst Haeckel in the 19th century to produce plates that are still reproduced as examples of natural beauty. Each one of these microscopic skeletons that falls to the bottom is a capsule of information about the oceanic conditions of the time in which the organism that built it lived. Abyssal sediment columns are, in that sense, archives of the climatic history of the planet that goes back tens of millions of years. Molecular biology techniques applied to the analysis of environmental DNA, or eDNA, are transforming the rate of discovery. Instead of requiring the physical capture of organisms, eDNA allows the detection of the presence of a species through the analysis of traces of genetic material left in the water or in the sediment. A single water sample of 10 litres, taken at great depth, can contain genetic fragments of dozens or hundreds of species present in that area in recent days or weeks. Genetic analyses of abyssal sediments have identified close to 100,000 DNA variants in samples from around the world, 60% of which belong to organisms that do not fit into any known taxonomic group. They are not merely new species, they represent entire branches of the tree of life that still have no name or description. The goblin shark, Mitsukurina ostoni, is one of the few living representatives of a family with a fossil record of 125 million years, since the Cretaceous period. Its appearance is unmistakable. A long, flat snout called a rostrum that protrudes from the head like a shovel, pink or reddish skin due to the proximity of blood vessels beneath a very thin dermis, and a jaw that can project out of the skull at extraordinary speed to capture prey. The jaw is not fixed to the skull with rigid joints, but can shoot forward through a mechanism of elastic ligaments, a system called ballistic protraction, that, in this shark, reaches exceptional speeds and displacements. The protruding rostrum is covered with electroreceptors that detect the electric fields generated by the muscles of other animals, an adaptation especially useful in darkness, where vision has a very limited range. The unanswered question about this shark is why the rostrum has such an exaggerated shape, when there are sharks with equally efficient electroreceptors in much more aerodynamic forms. 125 million years of evolutionary history support that the advantage of that shape is real, although not completely understood. The goblin shark shares the abyss with the frilled shark, or Chlamydosalatus anguineus, another living fossil whose general morphology has not changed appreciably in 80 million years. It has 6 rows of trident-shaped teeth designed to hook into soft and flexible prey, such as cephalopods, and it can bend its body in a manner similar to a snake to launch itself at angles that would be impossible for conventional sharks. Females have the longest gestation period known among vertebrates. In laboratory conditions, it has been recorded that embryos develop for 3.5 years before being born. In the depths, where temperature and metabolism are low, that period could be even longer. This extreme reproductive slowness makes these species especially vulnerable to any disturbance of their populations, because demographic recovery would be extraordinarily slow, even under favourable conditions. Cold seeps are areas of the ocean floor where seawater mixed with methane, hydrogen sulfide, and other compounds emanating from the crust slowly rises through the sediment. Unlike hydrothermal vents, the temperature in cold seeps is similar to that of the surrounding seafloor, but the concentration of chemical compounds is sufficient to sustain chemosynthetic communities. Cold seeps have been found in the Gulf of Mexico, in the Arabian Sea, in the deep Mediterranean, and in multiple areas of the Pacific. The communities that inhabit them include giant clams with symbiotic bacteria in their gills and bacterial mats several centimeters thick that cover the sediment. Even stranger are the so-called hypersaline lakes of the seafloor, or brine pools. In some areas of the Gulf of Mexico, and the Mediterranean Sea, extremely dense and salty water that emanates from specific geological formations accumulates in depressions of the bottom, forming liquid masses that are between 3 and 5 times saltier than normal seawater. Being denser than the surrounding seawater, they do not mix with it, and they form underwater lakes with well-defined surfaces and shores. Animals that fall accidentally into a brine pool die almost instantly from the extreme salinity. And yet, on the edges of these lakes, and to a certain extent within them, microbial communities have been found that thrive under conditions lethal to practically any other known organism. Explorers who have visited them in submarines describe the experience of observing their surface as seeing the shoreline of a sea within another sea, with currents and miniature waves generated by the difference in density between the two bodies of water. The deep ocean continues to generate questions faster than it produces answers. Each new expedition, each new instrument, each new technique applied to samples of abyssal sediment reveals organisms, processes, and ecological relationships that had no place in any previous conceptual framework. The ecosystems of vents, the biochemical mysteries of hadal fish, the gigantism of abyssal crustaceans, the parasitic reproduction of anglerfish, chemosynthesis as an energetic base alternative to photosynthesis, the omnipresent bioluminescence in the darkness, the 100-million-year-old active bacteria in the sediment, the unidentified sounds that travel thousands of kilometers in the water. Each of these phenomena opens windows toward an understanding of biology that surface environments cannot provide. Cold water coral reefs are another ecosystem of the deep ocean that has received increasing attention in recent decades. Unlike tropical reefs that depend on symbiosis with photosynthetic algae, corals such as Lophelia pertusa and Desmophyllum pertusum build complex calcareous structures at depths between 200 and 1,000 meters, at temperatures between 4 and 12 degrees, and in total darkness. These reefs grow much more slowly than tropical ones, on the order of millimeters per year, but they reach kilometer-scale dimensions when they accumulate over thousands of years. They harbour enormous biodiversity of crustaceans, fish, echinoderms, and mollusks that find refuge and food in their three-dimensional structure. Ocean acidification, produced by the absorption of atmospheric carbon dioxide, dissolves the calcium carbonate of their skeletons at a rate already measurable in some of these reefs. The submarine volcano Loihi, known to Hawaiians as Kamaihuakanaloa, the red child of the depths, rises 13,000 feet from the floor of the Pacific, 57 miles southeast of the Big Island of Hawaii. Its base, at 17,000 feet of depth, hosts fields of bacteria that feed on iron and excrete iron oxide, piled into dunes up to 2 meters high that cover acres of volcanic bottom. On its slopes there are formations of pillow lava, coiled and twisted like the wax of giant lava lamps, and hydrothermal chimneys whose presence has not yet been completely mapped. Each new submarine volcano studied in detail reveals an ecosystem with its own characteristics. The Denmark Strait Cataract between Greenland and Iceland is the highest and most powerful waterfall on the planet, with 11,500 feet of drop and 100 miles of width, and it lies 2,000 feet below the surface of the ocean. These are environments that most people will never know exist, but that are part of a living, dynamic, and incompletely understood world that regulates the chemistry and climate of the planet. The 5 Deeps expedition, the most ambitious scientific and exploration initiative in deep waters of recent decades, mapped 212,000 square miles of seafloor in high resolution, identified 40 new species, collected more than 100,000 biological samples, and demonstrated that in each of the deepest points of the 5 oceans there is life. The scientific team, led by Alan Jameson, responded monosyllabically at the Royal Geographical Society when a member of the public asked whether there would be an ocean full of life in 50 years. He stepped onto the stage, took the microphone, and simply said, yes. The certainty of that answer is the synthesis of decades of exploration in the most extreme environments of the planet. The abyss is not the end of the ocean. In terms of biological knowledge, it is a beginning that is far from being completed. Each new technique, each new vehicle, and each new expedition expands the map of the unknown at the same time that it reduces it. 95% of the ocean remains unexplored in detail, which means that the vast majority of its inhabitants still have no scientific name. Their ecological interactions have not been documented, and their contributions to global biogeochemical cycles have not been quantified. The magnitude of that ignorance is not an argument for hopelessness. It is an argument for the continuation of exploration. The oceanographer William Beebe, who in the 1930s carried out the first descents in a bathysphere and was the first to describe, in his own words, the beings that inhabited the deep ocean, wrote that anyone who had seen the abyss and returned able to speak of it with astonishment and with a deep understanding of the wonder of what had been seen would be forever changed. Since then, every person who has descended into that world and has found the words to describe it has expanded what is possible to imagine about life. The marine biologist Sylvia Earle, who has spent more than 6 decades studying the ocean and has carried out thousands of hours of diving in all the seas of the planet, refers to the ocean as the planet's greatest asset, and has repeatedly pointed out that the inability to see the deep ocean with one's own eyes has been the greatest obstacle for people to value and protect it. Intellectual awareness of its importance has not prevented its gradual degradation. What changes the relationship with any environment is direct experience, the sensory impact of being present within it. Deep-diving submarines, from the first military bathyspheres to modern titanium vehicles capable of descending to any point in the ocean, are instruments of knowledge, but also instruments of connection. Every person who has descended into the abyss and returned has described a transformation in their understanding of what it means to be alive on this planet. The scale of the deep ocean, its geological antiquity, the diversity of its life forms, and the radical nature of the adaptations it has produced are arguments for a specific humility, that of recognizing that most of the planet's biosphere operates according to rules that we have not yet finished learning.